Thank you for joining us for the 5th Annual Clinical Diagnostics and Research Online Conference. Life Technologies, a brand of Thermo Fisher, is a proud sponsor of this conference where we will focus on presentations themed around unraveling the complexity of oncology research. My name is Carol Koch, and I am Senior Marketing Manager of Clinical Research and will be your moderator for today's presentation. I am proud to introduce Dr. Jeffrey Weitzel, who will be our first speaker in this week's series. Dr. Weitzel is the Director of Clinical Cancer Genomics and Director of Cancer Screening and Prevention at the City of Hope Comprehensive Cancer Center. He's also a Clinical Professor of Preventative Medicine at the University of Southern California School of Medicine. Today, Dr. Weitzel will speak on establishing an economic BRCA1 and 2 gene analysis research workflow using custom AmpliSeq panel design and Ion PGM system. At the end of the presentation, I will be facilitating Q&A, so feel free to submit questions on the screen during the presentation. I will now like to turn it over to Dr. Weitzel for his presentation.
All right. Well, good morning. Thank you for joining us today. I see we have a number of attendees. First, a couple of points. I'm a medical oncologist and a geneticist, so I come to this from the perspective of being a clinician who does cancer genetics care and also cancer genetics research in my laboratory. So I really come with a clinician's perspective, though, and that's looking for things that are useful to me or useful to other people. And so you'll see when I do my presentation today that rather than it being, although there are going to be technical details and I'm not a technical virtuoso, I'm much more interested in how we use these tools to get to clinical care and the research along the way that's necessary to give us tools. So that's my perspective. Second is, I tend to pronounce the gene names BRCA1 and BRCA2. I spell them out, so to speak. The term BRCA really comes from the marketing of the one vendor who had single ability to market this test for the more than the last decade. So there's some in the field who are sensitized to the way the genes are labeled, but nonetheless, we use the formal gene names in any event. And I've come representing not just me and the City of Hope, but an entire clinical cancer genetics community of practice. We've got a training program for people who are trying to become clinicians and do work as clinicians in cancer genetics. And through this course, we have an entire community of practice in many states in the US as well as globally. And so I represent them here today as well. All right. So first, we'll start with, since we're talking about the BRCA genes, this is a slide from New England Journal of Medicine in 2010, but highlighted a rather interesting set of events. And it really relates to intellectual property and genes, and the essential thing being, can you patent a gene? Can you patent nature is a better way to say it. I've highlighted at the beginning here that you've got, for example, in 1953, the original publication by Watson and Crick. I like to highlight that because in 2003, it was rather fun to be talking about the 50th anniversary of their paper. And if you've ever read that paper, you really, if you haven't, you should. It talks about things in much more flowing prose than our usually very sterile discourse that we have in any of our given journals today. And they postulate about how this code must help to convey the information in a reliable way. And of course, we have a world of information since then. Then fast forwarding to the next highlighted box, in my mind, one of the critical issues was that there was an International Breast Cancer Linkage Consortium, the BCLC, that was founded in around 1989. And this was basically a set of about 200 very high-risk families that looked like hereditary breast and ovarian cancer. And they were, it was in this setting that they used as a common set of patients to try and do the linkage. So as we go forward, in 1990, the critical event was Jeff Hall and Mary Claire King's lab had done linkage analysis and showed that BRCA1, first off, that it existed. They gave it the name, BRCA1, breast cancer gene 1. At the time, Mary Claire King has commented on the fact that, she was at Berkeley, California then, and she jokes that BRCA1 stands for Berkeley, California 1, but the fact is that this was breast cancer gene 1. That's how it was named. And the linkage established that it existed. Up till that point, nobody believed that any single gene would account for a large proportion of cases. And second, gave us the address on chromosome 17. Then there was about a 4-year horse race of sorts, and then an academic industrial alliance resulted in the cloning and sequencing of BRCA1 in 1994. And that was reported at the Montreal American Society of Human Genetics meeting. That was my first human genetics meeting and was really quite an exciting time. I thought all the meetings would be that exciting, but of course they aren't always that exciting. 1 year later, because what was clear was that among the families, those high-risk families that appeared to have hereditary breast cancer, only a proportion, maybe 2/3, were explained by BRCA1. And so the search continued, and within the year, BRCA2 cloning and sequencing was reported. BRCA1, of course, is on chromosome 17, BRCA2 is on chromosome 13. Commercial testing in the United States first became available in 1996, and then this highlights the fact that patents were awarded by the U.S. For BRCA1 and 2. So at least in my world, we've had no choice but to use one given vendor that had intellectual property rights in this regard. Now, going forward, I highlighted here on this chart that in 2006, finally, will be the word I'll use, large rearrangement screening was added to the test. And I think that's important. As geneticists, I don't know if anybody who's attending this webinar who wouldn't normally want to be as thorough as they can for a testing strategy. And there's a clear recognition that for BRCA1 and BRCA2, up to 10% of mutations can be large rearrangements, which are not detectable by sequencing in general, at least certainly not Sanger sequencing, and that the number of large rearrangements, however, is really focused on BRCA1. The BRCA1 gene is 41% allo sequences, so it has a propensity to these large rearrangements, shuffling of the exons, either deletion or duplication. And so it's about a 5-to-1 ratio of BRCA1 rearrangements compared to BRCA2. So many clinical approaches often involve screening for large rearrangements at the least in BRCA1, sometimes ignoring BRCA2, though among our consortium that I'm going to tell you about, there is a BRCA2 founder rearrangement, I believe, that has to be detected by my colleagues in Europe. Then the next step in this timeline really is quite interesting, and that was this past June 2013, the U.S. Supreme Court ruled that as nature, genes can't be patented. And that's really opened up quite an avenue for the opportunity to take different approaches to genetic testing. So I'm going to highlight where we go from here. The other thing that happened, of course, almost at the same time, I think a month or so before the Supreme Court decision, was Angelina Jolie's announcement that she was a BRCA1 carrier. This really did put things back in the spotlight, even though I thought that clinical cancer genetics was getting a bit staid. We've been doing genetic testing for BRCA, at least in the US and Western Europe, for more than a decade. And over that decade, acquired information about what we do with individuals. But Angelina really reminded us that no matter how much we think we've been teaching about a given element of what is important in health science, that such publicity can really magnify. And so, again, threw it into the spotlight. For those of you who don't know, again, this is publicly available information. Angelina Jolie's mother died of ovarian cancer at age 48. And ovarian cancer is one of the sine qua nons of hereditary breast ovarian cancer syndrome. Now, this is the actual sequence of BRCA1 and BRCA2, and I want to credit for this slide, of course you can't read it, I want to credit for this slide Tom Frank, who was the first laboratory director for Myriad Genetics, and he was a wonderful man. He has passed, but he's a wonderful guy, and he had shared this, and of course you can't see it, but in this case there was an insertion of an A that shouldn't have been there. And so going through this used to be quite difficult and expensive. Expensive meaning in the US, it started at around $2,000 for complete sequencing of these 17,000 base pairs of coding in adjacent intronic regions. And it most recently was as much as $4,000 US to completely analyze BRCA1 and BRCA2. And now things, of course, have moved on, and we'll talk ever so briefly near the end about multi-gene panel approaches. But this was the state of the art for the longest time, and this is the actual coding sequence. And I'm using the past tense here about was difficult and expensive because, of course, we're going to be talking about next generation technologies here. So among the next generation technologies, and I would refer the listener to the available review, this Journal of Clinical Oncology in 2013 had a series of articles, the reference is at the bottom here. There had a series of articles that were really quite helpful as a summary of the clinical applications of genetics and genomics. So I would refer the reader there for further information, but it really highlighted the fact that you've got Sanger sequencing, which has been the gold standard, fairly expensive on a per base pair perspective, but really high accuracy and considered more or less the gold standard. And when you look at the traces, typically we're suppressing anything that's below a certain level and therefore trying to maintain high read fidelity. One of the things that's interesting that we don't have time to talk about today, but is relevant to next generation approaches, is that with this massively resampling, again, the massively parallel sequencing, which is in the next two boxes, and the next box in particular, we can start to see mosaicism in some cases. That is, small subpopulations. That's, of course, one of the advantages when applying these tools to somatic genetics, that is, the genetics of a given tumor, looking for druggable targets. And we're often looking at minority populations. So you don't necessarily suppress the reading of the minority populations when using the massively parallel sequencing. But, of course, this massively parallel approach comes with economies of scale. So read accuracy goes down, but the cost per base pair goes way down. And that's really been, and then I think some emerging technologies are very interesting. Single molecule analyses, again, slightly lower read accuracy, but extremely low cost. So I think this is a very exciting time to be involved in genetic and genomic research because of the potential for these differences. And the cost differential is depicted here. For those of you who aren't familiar with Moore's law, Moore was involved in semiconductor science. So I believe he was one of the founders of Intel. And his theory, and what really was borne out, was that basically something like every 18 months they can double the number of transistors on a silicon chip. And that increases the power and decreases the cost. And so that's why our phones today have more computer power than the rocket that went to the moon a number of years ago. So reality is that the semiconductors have been getting cheaper every year by the megabyte. And for quite a time, genetic technologies and genetic sequencing costs followed the same line. Until recently, it just completely dropped off. So around 2008, when the next-gen sequences were first introduced, you really see this huge drop-off in the cost. And I think the reality is here that, at least in terms of raw sequence read, it is possible now to do the entire genome for about $1,000. Now, not necessarily a good fidelity depth of reading, and doesn't include the bioinformatic analysis, but about $1,000 to acquire the data at a rough first pass. And I think that that's pretty remarkable, given that up to the time the first genome was reported around 2001, it took 10 years and more than $3 billion to sequence one person's genome. And now we're talking about doing the entire person's genome in perhaps a couple of weeks for perhaps $1,000. Underneath is the different technology platforms that have been advanced. Pyrosequencing was quite exciting when it first came out in 2005, but I think it's really been supplanted now when you go further with many of the Illumina machines. And of course, we're talking about Ion Torrent and the life sciences products here today as well, but the Ion Torrent PGM, as well as the proton and other systems. So these are really the more current systems in use, and I'll talk a little bit about our use of at least one of those systems. So again, I'm a clinician, remember, so my focus is on breast and ovarian cancer. It's actually on all hereditary cancers, but for this talk's purposes, breast and ovarian cancer. And the other thing I do a lot of work in is the realm of disparities. And in disparities, I mean, who has access to genetic testing? So for the most part, it's only been Western European and other individuals, but the question is hereditary cancer different in different populations. At least 5% of breast cancer and 15% or greater of ovarian cancers are hereditary. Of course, you end up having to test 10 times the number of people who have a mutation to find a mutation if you're following guidelines. That is, you'll test 10 people to find one BRCA carrier, and for the ovarian cancer, maybe test 5 people to find one. You do see these small proportions. So it's really, the problem is breast cancer is a common disease, so figuring out who to test is one thing. If the test is cheap enough, you just test everybody. Now, to get to this question of other sizes. So again, this is Soraya. She won a Latin Pot Grammy. And I'm not presenting personally identifiable health information. The pedigree that you see on this slide was derived from her obituary. Her mother, her aunt, and her grandmother all had breast cancer. So I think it's a crime that this person developed breast cancer locally advanced at 37 without any real warning or opportunity for predisposition testing. And so does it occur? Do genetic causes of cancer occur in Latin America, for example, one of my areas of interest and focus? Yes, it does. So as I mentioned, access to care influences the knowledge of genetic epidemiology. Almost all of the world's literature on the genetic epidemiology of breast cancer comes from the United States, Western Europe, Iceland, and Israel because of unique founder effects. And Australia has a very well-developed cancer genetics system as well. Now, other parts of the world, Colombia, we've been doing some work, and others have reported there. The Bahamas have some unique genetic features in terms of founder effects. And my dear friend and colleague from the University of Chicago, Dr. Fumio Lapati, has done amazing work in Nigeria. And I know that Dr. Tim Rebick, when it comes to prostate cancer and other genetics, has been heavily involved in Africa. So many of us are devoted to the cause of bringing genetics to all world populations with the idea of enhancing access to this care. Now, the North American commercial vendor published in 2009, Michael Hall, who's at Fox Chase University, was the lead author of this paper summarizing Myriad's data. And just goes to show you that if you look at this panel, that Western Europeans were the majority of cases that were sequenced, followed by Central Europeans. Interestingly, the Latin American population here, almost 2,000 cases when published in 2009, but I can tell you that my program alone, because of our outreach to the local Latino population at one of our county safety net hospitals, was responsible for 500 to 800 of these cases alone. So a substantial portion, but still a pittance if you look at this, less than 2,000 here compared to 20 times that many people from Western Europe. And so the consequence has been very little knowledge about many of these subset groups of individuals. So let's see here. I'm like looking for my pointer, but oh, there we are, edit. Nope. I want to edit. I'm just trying to find my editing wand, but that's okay. So I work at City of Hope. I'm out in Los Angeles. This represents a map of my collaborative network, people who are working with us to try and solve genetic epidemiology issues. These are people in my community clinical cancer genetics community of practice who are employing a common protocol for our registry. So this is just to illustrate that. But what I wanted to highlight is that I have collaborators in Guadalajara, Dr. Adrian Tineri and Azucena del Toro, and at INCAN in Mexico City. So Cynthia Villarreal, Luis Herrera, Alejandro Mojar, Rosie Alvarez. There's a whole team of people we're working with. And in Puerto Rico with Marcia Cruz Correa, Julie Dutil, and Dr. Umpierre. And so folks across the country. And together, by looking at the epidemiology of BRCA in Latinos, I published a year ago on 14 network sites, 746 participants, a large number of mutations, and we were able to generate some hypotheses. But I wanted to point out that, again, this was the largest population of Latinos sequenced for BRCA genes in the literature to date in 2013, given the representation of nearly 40 or 50,000 for Western Europeans. You can see there is still a disparity. So we joined together This was a group that was more or less self-assembled, led in large part by Jose Luis Costa and Jose Carlos Mercado of Ipatinib in Portugal, and by Dr. Mezenkamp and Lichtenberg in the Netherlands. And also, I've been pushing folks at Life Sciences to help us move this along as well. So between us, we were convinced that developing a common platform for doing BRCA testing on a next generation platform that could easily be disseminated was worthwhile. And so that's why I coined this deploying a common protocol. So this may not look as global as it could be. Obviously, we're missing Asia, Africa, and South America, but most of what we do will be applicable to those other areas. So I wanted to highlight Dr. Hidalgo, who's at the INMAGEN, the Genomics Institute in Mexico City, our center in Los Angeles. We have a center in Canada, Scotland, and of course Portugal, the Netherlands, and also of course in Germany. What was the consortium's goals? One, to develop an AmpliSeq platform for doing next-gen research with BRCA genes. Ultimately, we'd like to get the technology up to speed and the informatics pipeline so that this could be applied where possible for benefit of the individuals. But our research encompasses the following. First, we need a good coverage of the target. If you're going to try and do clinical grade sequencing, you need 100% coverage of all the coding exons and intron exon boundaries, plus 20, minus 20 generally. Applicons covering the exons need to be overlapping, and we need to be careful about that in particular because of the potential for SNPs, single nucleotide polymorphisms, to disrupt any given amplicon. Wanted it here, given that it was being developed initially by the folks in Portugal and the Netherlands, to the European Molecular Genetics Quality Network Guidelines. I'll comment on that again in a moment. And then we want it to be adoptable by other research labs. Accurate, affordable, and easy. Easy is a word abused often, and I think that applies to any new technology. But I will say that the ability to do a single day or a few day workflow to do a certain number of chips, and actually this is a mistake here. It says at least 6 samples per chip, and you'll find out that we have up to 8 samples for the 316 chip currently. And again, we want it to be reliable with easy data analysis. For this purpose, you can have many different workflows. Of course, the Ion Reporter is associated with the Life Sciences group, and so if that could be a turnkey system, that would be ideal. So the resulting design was felt to meet requirements. So the folks in Portugal and the Netherlands got together, working with Life Sciences, designed the pool, 167 amplicons, 3 primer pools, requires 30 nanograms of DNA starting DNA, which is really a minuscule quantity, of course. There were 200 base pair designed because the PGM, the personal genome machine, the Ion Torrent, can get up to 200 base pairs fairly reliably. That differentiates it from some of the other platforms where perhaps the read lengths are shorter, 100, 150. Formalin fixed paraffin embedded, sorry for the acronym FFPE, can be done, but slightly lower performance because of the poorer quality of the DNA, but it certainly can be done. I think the proton's probably more adept for that. And again, no SNPs in the 3' end of the primer. So these are the European best practice guidelines, which are highlighted in the blue box here. We started with a 3-phase project plan and verification experiments. First phase was involvement of the design, which I just described for you. Next was to analyze 20 archive samples, and this was again done between the Netherlands and Ipatinib in Portugal. 9 MER homopolymer variants were particularly focused on, as well as a look-see to see if we could see large rearrangements. That's what the MLP variants are referring to. The flow is fairly straightforward. Multiplex PCR with 3 pools, 55 primer pairs. Barcoding is, of course, done to enable the multiplexing. Template is prepared. You can see the time listed there. Again, these are idealized times, I would have to say. You got the barcode adapter ligations. You run the sequence, amplify, and analyze the data. So this is the idealized sequence that you can see the idealized hours here. So it is technically conceivable to do it in a day's run. Though I think in practical terms, my technician, Joseph Herzog, who's very adept at these things, in fact, incredibly adept at these things, might take it 2 days to get it done. But again, with meticulous care, you'll get good results. Also, it's learning the protocol. The first runs, this is the data from Patinib and from the Netherlands showing the metrics were good. They had 100% coverage of the open reading frames. There was average coverage uniformity of 98.8%, which is pretty good, average on-target specificity. I am not a bioinformaticist, and so you'll have to judge the individual characteristics yourselves, and certainly would encourage you to consult with the folks either at Ion Torrent or otherwise regarding what this means in terms of the quality, but this is high quality. Fine, so got through the design. Now 30 archive samples with 65 different variants tested, exchanged across 2 labs. And so they were able to look at variants and were able to, of course, compare between their 2 labs that they could each pick up the same things, deletions and insertions, point mutations, and then they tested to see if they could pick up exon deletions. They multiplexed 8 samples on one 316 chip, and from there, it's hard to read this, but the pipeline really just says that basically this is a standard pipeline. The assembly of the alignment, the coverage analysis, SNP and NDEL detection, annotation of the variants, then the ability to interact with the various databases to identify known pathogenic variants, things that are already in the dbSNP database as polymorphisms and the like, and to verify these variants and then to extract the report. So this is the Ion Reporter depicted workflow. Others have applied their own workflows that have of their own. But I think this is a workflow that we were all trying to use as a consortium, looking for a turnkey operation. So the report may look like this when you get it done. And again, here, this is just highlighting. You can't see it very well, and I can't amplify it. But basically, you have the identifiers. This will tell you which individual. This will tell you the type of change. This is the predicted protein coding change here. It's a 4 base pair change. And then you've got it's the gene is BRCA2. And then you can get over here to get some of the annotations. And does it have an entry in dbSNP? Oncomine type. And so these are the kind of annotations you can get. There's a real learning curve to this, but you can customize your own workflow. If your unique population has recurrent mutations or recurrent variants, you can ultimately filter this in different ways. So I'm not going to go into the details of that because it is really use it and figure out how to do it sort of territory, but suffice it to say that you can customize the workflow. And at first, I will tell you, as a non-bioinformaticist person, it is daunting to me, but others can certainly manage it just fine. And it's exciting to start combing the data and seeing what you can get for yourself. Reproducibility studies were done. So in this consortium that we created among all the sites, almost all the sites, so here's the City of Hope, that's what COH stands for. The other sites are mentioned here. So there are certain variants, the ones that are listed here, that were selected and tested across multiple site platforms to be sure that we could all detect the same things. So again, multiple layers of overlap and non-overlap to be sure that we had sampled thoroughly. Only a few spots didn't have those mutations in their pool. So this is a summary. We've had 100% coverage of the target regions. The minimum coverage was 100x. And that was the goal, was at least 100x coverage. I think the standards for sort of clinical grade sequencing are still in evolution, but most people feel that at least 75 to 100x coverage is essential. You can see here that although it is hard to read the numbers, that we're literally at 1,000x coverage for most of the exons. And these are multiple different labs pooling of their runs to show that pretty reliably across multiple labs, we can detect a high level. One Amplicon had as low as 60x coverage, and that was the exception. We've done some subsequent spiking experiments, we, meaning the folks in Portugal and the Netherlands, in terms of their respective labs. They really were the ones who were doing the powerhouse initial development of this, and are trying to rectify that, but really it doesn't need to be rectified. It doesn't have a lot of homopolymers in that read area, and we've saturated that area with additional mutations to detect to be sure that it was effective. Nonetheless, good coverage. again, you can see that variants can be detected in this exon with the current workflow. 350,000 mapped reads on average per barcoded sample. So for a gene of 17,500 coding sequence, that's pretty good coverage. Mean depth of coverage, as I mentioned, was over 1,000. So here as much as 2,000 per amplicon. Thus, we had done this, and then the next step was global verification. So we've got good performance characteristics. Can we, among our 8 labs, find all the variants that we think we should? So we had archived samples that had known complete Sanger sequence variants and that were well characterized. So each of us selected lists that were non-overlapping, so that there were some that were overlapping because they're seen globally in different settings, and others that were unique to given countries. And therefore, we achieved the opportunity to have, I believe it was 151 unique variants. We also had to work with the false positive detection rate because we noticed, for example, that large deletion, that was a del56 mutation, was not being picked up. It was being suppressed in the bioinformatic pathway. Well, we then adjusted the software. This is Chrysanthi, who does most of this work for life sciences, and really worked hard at creating the flow, the workflow. But you can see here the true positive, false positive rate. Very good numbers here, although some of us might argue that if you were trying to apply this in the clinical setting, you'd have to have improvement on this because you have to be able to be sure of what you have or not have to do a lot of secondary verification steps. Certainly for now, one would probably want to do Sanger sequencing of a given variant to confirm it. And there's a lot of other reasons to flow in your clinical realm. I won't comment on that. I don't run a clinical laboratory. I run a research laboratory. But others who are interested in this pathway are interested in ways of designing economical workflows for commercial purposes. Nonetheless, this is using 8 samples on a 316 chip. This is what our consortium has worked on together. I can tell you that using the larger chip, the 318, you can get even greater economies of scale. And we scaled up the number of samples proportionate to the capacity of that chip. And again, just the complexity with the barcoding is quite manageable. So this is, again, a summary of the predictive values. So in long homopolymer tracks, there were 11 unique mutations. We really made sure that there was good representation of mutations in those tracks, which are known to be challenging for calls. 12 of 13 were picked up, 1 no call. The rest, and there were no missed calls. There was 1 no call, so at least you'd know to look there more carefully, with a calculated sensitivity based on 12 to 13, 92+%. For indels, 120 of 120 picked up. Again, no missed or non-calls. And then for point mutations, excellent coverage, 86 of 86 picked up. So the overall, across all of these, even the challenging stretch bits, was 99.5% sensitivity. I don't think there's any technique out there that has 100% sensitivity, but I think that would have to be considered. So the problem is of course, not all of us have a bioinformaticist. I love the New Yorker articles or the New Yorker cartoons. It was so typically brilliant of you to have invited a bioinformaticist. This actually used to say epidemiologist, but I changed it because I think that bioinformatics is an art that we need even more today to help make smooth workflows. So, in summary, our global consortium, really a band of brothers and sisters, have been using this technical platform. It's been established in all of our centers, and I think has excellent performance characteristics. 100% coverage of the targeted regions, more than 100x coverage across all but 1 amplicon, and some of that's being dealt with. Mutations detected were in homopolymer stretches, deletions and insertions, point mutations, exon deletions. Now, reliable large rearrangement identification is much more challenging. You actually need to have primers that tile across introns to really get good breakpoint identification of amplifications and duplications and deletions of exons. We are able to see by pooling a number of samples in the one run and then using them as internal controls against one another, we were able to see one of our large rearrangement mutations that's common in Latin America or in Mexico, but not as reliably as we'd like to. So there are still efforts in progress here to try and create CNV detection methods to use this same workflow to get there, but I think it's going to require some additional modifications. As mentioned, barcoding enables analysis of 8 samples simultaneously on the 316 chip, and we can do more on the 318 chip. And again, this is a demonstration that as a group, with close collaboration among peers, this work demonstrates the potential for mutational analysis of BRCA1 and 2 using the Ion AmpliSeq technology combined with the PGM system. One of the reasons I chose that system didn't have to do with it being the cheapest system, because frankly, on a base pair by base pair basis, it's not the cheapest system. Much larger platforms are cheaper, but you have to barcode in and multiplex multiple samples to get those economies of scale. I was also looking for a platform that didn't have a high cost for the equipment itself, because my interest, as I started at the outset, was to try and create a protocol that can be deployed in low and middle income countries in areas to democratize access. And so this particular platform, at least on the cost for hardware, is the cheapest platform of the next-gen sequencing technologies. I think that's a reasonably verifiable statement. And so that meant that it was probably, in fact, it's cheaper than a typical capillary sequencer of the Sanger type. So I think that that's important. And then the feasibility of deploying this common protocol to diverse geographic settings, I think, is established by our group. We have a manuscript in draft at the moment to describe our experience with this. But I will tell you that it is feasible, but it still requires technical virtuosity to be sure you're getting all of this right. And I think that the further refinement of the bioinformatics pipeline, and I think as people learn the protocol, I think, and there are good measures of quality, I think there's a good opportunity to get what we need out of this. Now, the other thing I want to highlight is that I'm interested in moving beyond BRCA, of course, but we have to take it one step at a time. This is a paper that was originally by Dr. Stadler, where this table was created. Many have seen this table in different settings. In the upper left are the high penetrance genes, the genes that are really clinically relevant and have management to go with them. BRCA1 and BRCA2, the colon cancer mismatch repair genes are there, hereditary gastric cancer from the CDH1 gene, Li-Fermani, and the P53 gene. So a lot of us would like to see multi-gene panel approaches, and I think these platforms are adaptable to that sort of approach. In the middle is our biggest challenge. There are many moderate risk genes that we aren't as much certain about how to apply them, but they're nonetheless being incorporated into panels. In the bottom right are the common but modest risk variants, which you can get from deep sequencing and other ways of doing this, but I think that they're not as much clinically useful at the moment. So we're really focused on the upper left. We're adding the middle, and soon I think we'll be doing more overall. But this is a sort of a view to the future, and the current and the future, actually, as many of the vendors in the US and elsewhere have started to move to multi-gene panel testing. So hereditary breast and ovarian cancer affects all world populations. Sanger sequencing and screening for large rearrangements is the gold standard of BRCA gene analysis. I believe that economic next-gen platforms could be deployed to democratize access to full BRCA analyses and multi-gene genomic panels. I think the full BRCA analyses are probably more critical at the moment, and that's because if you look at a pie chart of hereditary breast cancer or ovarian cancer, 75 to 80% of the true positives are still those two genes, as compared to 10 or 15 genes to make up the next 10%. So I think we'll make a lot of bang for the buck if we go forward here. I want to acknowledge some of the funding for my Latino work, in particular the Breast Cancer Research Foundation, Avon Foundation, both critical support to our work in Latin America, previous NIH R03 support, and critical support from the American Cancer Society, both for my genetic epidemiology and my behavioral science work in Latinos. So I believe with that, I have to put up this legal last slide, so to speak, that this is for research use only according to official use. And I think everybody understands they have to get things to their own relevant definitions of clinical grade in their own setting, following their own local regulations. But I think that's the end of my presentation, and we'll now open it up to questions and answers. And what I'm going to do at this point is to share this. I want to thank you all for your attention, and I'm going to now turn over All right, so I guess I will have, unless Carol's able to unmute herself, I will have to do the Q&A for you. So let me go to some of the answer to the questions. So how long is it taking your team to do this entire project? And that, I will tell you that we've been working together for more than a year. We have a presentation at American Society of Human Genetics from last fall that is a poster that we're willing to share. Happy to do so. But we've been working together for almost 2 years. But I think the preliminary work by Mesenkamp and Costa, I think, was critical to getting that going. I think it took them probably better part of 6 to 12 months to get their initial testing done. Anytime you're trying to coordinate a global consortium, so 7 different centers, each with a different infrastructure, all of us getting our systems up and running, that's a challenge. So we end up having to take some time to get everybody up to speed and then to get everybody coordinated. We're all doing the same protocol at the same time so that it can be reproducible, and so we can basically represent that this should work for you too. So I think that's the idea. But it's been a couple of years, and we're really looking to the next level. So, we're really at the point of, okay, let's really refine this bioinformatics pathway, make sure it's doing what we want it to do locally, now that we know that we can get the sequence out of this machine, and what are the optimum parameters for spiking to improve that one amplicon that had slightly less representation, these sort of things. So that's what we're doing now. I'm going to hand back to Carol for the moment, because I think she said she's got her.
Great, thank you. So we had another question around what was the biggest need in terms of analysis softwares? What was really your needs for this program for that?
I think, yeah, there you go. Well, I think that's a good question, but I'm going to tell you that's really going to be more of a question for the individual labs. Many labs have been doing different pathways, different workflows, and there are a number of workflows out there that I'm sure anybody who's doing next-gen sequencing knows there are multiple different workflows. I think the issue here is that for our purposes, I'm looking for a system that can be turnkey, so my interest in seeing them get the Ion reporter to a point where it's really easy to use and annotated in a custom way for BRCA1 and BRCA2, and especially annotated for a given population group. So for Latinos, I want to get all of the things that I already know are variants, because we've been testing a lot of people. So we know many of the variants, and if we can get them annotated in the workflow, we don't have to keep recognizing what appears to be VUSs. We already know they're polymorphisms. And so part of it is customizing the workflow. And if you've got a single need, analyzing 2 specific genes, and you can customize the workflow to represent that, I think that works fine. That's my need, because I want other people who can do this to do it. So I want to be able to hand this technology off, or I should say the protocol off, to my collaborators in Latin America so they can do their own testing and their own sequencing. However, I would certainly defer to others who have much more experience in terms of the bioanalytic pathways about the merits of any given pathway. So that's been my biggest need, is just having something that's turnkey and can be customized. And I think probably it works for this way. Let's see.
Great, thank you for that. We have another question that wants to know, you mentioned that there were 30 nanograms of input DNA. Is that the amount that was used for FFPE samples, or was that for whole DNA? And is that the lowest you were able to go for a sample input size?
I need Carol to pick up.
Oh. Is it okay? We had another question from a researcher who wanted to understand about the input amount that you used, which is, you mentioned in the presentation, 30 nanograms. Is that the lowest amount that you were able to use? And is that the amount you used of FF?
All right, so I'll pick up. The next question that I saw was, to what extent is the deep sequencing information used for personalized treatment? That's an interesting question. At this point, we're not talking about, our consortium was looking at germline variants in BRCA to know how to do, if you want to talk about personalized medicine, let me tell you that clinical cancer genetics has been doing personalized medicine for decades, identifying high-risk families, doing risk reduction surgeries, doing enhanced surveillance. So we have data on MRI, we have data on risk reduction mastectomy, we have data on all-cause mortality reduction for oophorectomy. That is personalized treatment. We know what we need to do once we identify the high risk. Now, there are others who, of course, use next-gen sequencing to do deep sequencing of tumors and identify targetable lesions. So I think that certainly it's possible, and this platform can do that. The Proton, I think, because it's slightly shorter read lengths, is been optimized to some extent for formalin-fixed paraffin embedded. I'll take the next question in sequence here because it's related to the question before it, which is, you mentioned 30 nanograms of DNA. What amount was used for fresh formalin-fixed paraffin embedded? I didn't cite data from FFPE, and I defer to the technological support staff regarding that. I know the group at the Netherlands, Dr. Mezenkamp and Dr. I'll think of it in a moment. My colleagues there, Lichtenberg, I know that they published recently on the sequencing in colon tumors of the mismatch repair genes, because they were able to use formalin, and that is a published paper, so perhaps we can post that afterward, but that's a published paper using the ion torrent as a way of sequencing the tumors for mismatch repair genes. The intent there was, of course, to establish that it was an acquired somatic mutation instead of a germline mutation associated with a microsatellite unstable colon tumor. But they were able to successfully amplify and analyze the mismatch repair genes from formalin-fixed paraffin embedded tissue. I know that this method and this circumstance is entirely possible to do on formalin-fixed tissue. This, I believe, is exciting because we're actually doing some pilot studies now to see if we can detect germline mutations in archival specimens. And the reason is that you don't always have the affected person in the family to test. If mom developed breast cancer and died at a young age, if we have archival tissue, we may be able to use her as the as the check, so that we know whether our testing is informative. So, there are people talked about. There's some other technical questions in this queue about how much for the paraffin-embedded tissue analysis, but really not my forte here, so I'm not going to be able to address that directly. All right, so let me just see if we have any other questions that were not yet answered. So I'm excited to try more of these things, but just trying to get, I think that might be the last of the questions, if I'm not mistaken. Yeah, I think that's it on the questions. So anybody else, please, we still have about 80 people online, so do put forth any other questions you might have. We are reaching the end of our hour. I wanted to thank everybody. I don't know if, Carol, I'll turn it over to you to see if you have any other comments, but I don't see any other questions at the moment. Oops.
Great. Thank you, Dr. Weitzel. I would love to thank you for your wonderful presentation and for all of you on the line for participating. We invite you to join us for 2 additional presentations that are coming over the next 2 days. On tomorrow, we have a presentation from Emma Bowden. That's 7:30 to 8:30 Pacific Standard Time, and that will cover the bioinformatics session that will speak about identification of predictive biomarkers from high-throughput drug combination screening data. And then on Friday, we will cover a session again on oncology that is about gene fusions. And that will be at 7:30 in the morning until 8:30 in the morning Pacific time again. If you would like to learn more about ion torrent technologies and products available through Life Technologies, please visit our booth. This can be accessed from the exhibit hall through the home button on your screen. Thank you very much. And we'd had some questions about receiving the slides. If you would like to receive some of the content that was presented here, we do have a poster in that booth that covers the content that Dr. Weitzel covered today. Thank you very much.